Hello, everyone, and thank you for joining us for this year's 2023 installment of the Advances in Proteomics and Metabolomics online symposium. My name is Princess Senate, your event moderator, joined by my colleagues Elizabeth Bowen and George Hammond. Before we delve into today's talks, I'd like to share some important information and tips to help you get the most out of this event. We kindly ask that you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. While we encourage lively debate and discussion, we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, particularly our platinum sponsor, Biocrates. For detailed instructions on how to make the most of the features available to you at this event, please visit the event homepage and watch the overview video at the top of the page, or visit the frequently asked questions section in the left-hand menu. Now it is with great pleasure that we introduce our distinguished opening and keynote speaker for today, Dr. Michael Snyder from Stanford University School of Medicine, presenting a talk on transforming healthcare using deep data and remote monitoring. Dr. Snyder, a pioneer in precision medicine, has spearheaded groundbreaking technologies spanning systems biology, RNA sequencing, and protein chip technologies. His impressive publication portfolio of over 800 papers reflects his esteemed status in the scientific community and holds the distinction of being the first researcher to amass petabytes of data on individuals, a scale that is truly staggering at 1 million to 1 trillion times more data than the average clinician collects. And without further ado, I will now hand over to Dr. Snyder to begin his presentation.
Great. Well, thanks very much for having me here. It's a pleasure to tell you about the work we're doing using deep data to try and transform healthcare. So many of you may feel the same as me that the healthcare system is broken, at least in the United States. We really tend to practice sick care rather than healthcare. And even when we practice healthcare, it's very antiquated, to say the least. If you think about it, what we do is you typically will get a car or a bus or what have you to go to a physician. When you arrive there, you'll show up at a doctor's office. It pretty much looks the same as it's looked for the last 40 years. When you're there, they stick a needle in you, which usually hurts, and draw a very large aliquot of blood. And from all that blood and the time you're there, they actually make very few measurements. And then from those measurements, they'll treat you based on population averages. We think every one of these steps can be changed. And in fact, just as an example, with regards to this last point about treating you based on population averages, you've probably been told since you were little that your temperature when you put a thermometer in your mouth, at least if you're in the English system, it would be 98.6. That's something like 36 point higher, I guess. Or sorry, it's more like 37 in centigrade. But it turns out if you actually look at the data that's out there, the average is more like 97.5 or the median. But the more important point is there's a spread. This is actually the 25th quartile, 94.6. This is the 75th quartile, 99.1. And what that means in today's world is that if your normal baseline healthy temperature is 94.6 and you go to a physician, they measure you at 98.6, they'll tell you you're healthy, everything's fine. What are you doing here? Go home. But I guarantee if you're up 4 degrees over your baseline, you're not healthy. Something's off. And so that's a big theme of our work. Know what your healthy baseline is so you can detect shifts from that baseline. So your health, you probably know it's influenced by many things. Your DNA is clearly a major driver, but many other aspects, many other items influence your health as well. Your lifestyle, such as your activity, the food you eat, stresses, environmental exposures. We know these all impact your health. And we're in a world where you can now quantify a lot of this, some of it pretty well. You can quantify your activity with wearables. We'll talk about that. Food is still a bit clunky, but you can do that too. Stress a bit indirectly. And these, we've set up new methods for being able to quantify environmental exposures. Even if you don't quantify these directly, you can quantify their effects by what they're doing to you, what they're doing to your biochemistry and physiology. And thanks to a revolution in technologies, we can now measure many, many things. We can sequence people's genome and measure all their RNA. at fairly low cost. In fact, it's estimated to cost $100 by the end of the year to sequence someone's genome. Mass spectrometry lets us measure hundreds of molecules out of people's, or thousands of molecules out of people's blood and urine. Wearables let us measure physiology. And so a number of years ago, we set up a project to be able to better follow people's health, if you will. They're using these advanced technologies I just described. Basically, what we did was you start with a small cohort, roughly 109 people. I'm one of them. And we'll actually take their blood and urine, saliva, stool, and other things. And we'll from the various parts, we'll sequence mostly blood. We'll sequence their genome. We'll even measure their epigenome, not shown here, their RNA, their proteins, their metabolites. These are all out of peripheral blood monocyte cells, so your immune cells. Proteins and metabolites will measure also out of plasma, measure lipids as well. We also follow the microbiome out of stool and other sites as well. This is on top of wearable data that we collect. We really do deep clinical testing, questionnaires, and even some advanced tests like stress echocardiograms and glucose control measurements. And in the end, we literally make billions of measurements on people. So one aspect is to do very, very deep data collection. And the other aspect is to follow people over time. We'll profile them longitudinally every 3 months while they're healthy. And if an adverse event like a viral infection comes along, we'll take 5 to 7 additional samplings. And we've been running this study for over 10 years now, 10 and a half, in fact. You might say, why are we doing all this? What's the point? And the answer is we're trying to understand what it means to be healthy. How does it change over time? What's the difference between different individuals? What happens at the earliest times of infection when you first get sick or in strange environments? And lastly, and very relevant to today's healthcare world, is we want to know whether these advanced technologies like genome sequencing, wearables, things like this, can they be better used to manage people's health? And I can tell you when we started at the time, it was quite controversial. And the answer with regards to this last question is just from the first 3.5 years of studying 109 people, we had 49 major health discoveries, meaning half the people learned something pretty important about their health. And it spanned a wide range of areas: hematology, cardiovascular disease, metabolic disease. And some of these turned out to be a big deal. We caught someone with early lymphoma, 2 people with pre-cancer, 2 people had serious heart issues, 1 through genome sequence, 1 through wearables. And I want to emphasize all these were found pre-symptomatically or without asymptomatically. The people did not have any symptoms whatsoever when we picked this up and different technologies and caught the different things. So for example, genome sequencing found one of the serious heart cases. Wearables caught the other one. Turns out imaging with some biomarkers found the early lymphoma. These were picked up through biochemical markers. So again, no one technology found this. Different, usually combinations of technologies revealed what was going on. And the way we like to think about this is that if your health is a 1,000-piece jigsaw puzzle, we're trying to measure 500 or 600 pieces, get a much, much clearer picture of people's health rather than the 5 or 6 they do today, which we think is woefully inadequate. So this is basically what we do. Just to give you a feel for this, we do quite a bit of genome sequencing. From the first 70 people, basically 12 learned something pretty important about their genomes. This is what you call Mendelian cases of single gene mutations that with high frequency can lead to mutations. So specifically, we found someone with a BRCA mutation, which puts them at high risk for breast cancer. So it turned out to be a male. We had 2 people with the mutation in this gene. One of them turned out they did a whole body MRI. It puts them at high risk for certain cancers. They did a whole body MRI follow-up. And once you know it, they had early thyroid cancer, and that was removed. And they're in good shape. We had 9 people who thought to be diabetic. One of them turns out to be actually a MODY mutation, wasn't really a type 2 diabetic as was thought. They're a different kind of diabetic, and they've been on the suboptimal medication for years. So the point out of all of this is that, genome sequencing was a powerful tool. It's on a minority of cases, only 12 out of 70. But if you're one of these 12, it's a big deal. In some cases, we think life-saving to find this stuff. I will, just for an aside, let you know that I know it's proteomics meeting, but we're big on all kinds of data, and including genetics. And we've come up with new ways of analyzing genomes using AI. And basically, using this method, we can always find 10 to 100 times more genes by looking either at rare variants or mapping what's called GWAS signals into open chromatin regions. And I can tell you, just in some interesting cases like ALS, where 7 genes have been identified by what's called genome-wide association studies, we found 700 genes and went from 6% of the heritability to 36%. Likewise, for severe COVID, we found just over 1,000 more genes. And so these new AI methods are very, very powerful for dissecting the genetic basis of disease. And we can even set up, for the case of AA, predictive models based on a genome sequence alone works. Genome sequence with EHR works even better. So one of the things that's of kind of high interest is that because we're profiling people over time, we can actually see how they're aging. And we found 600 molecules that track with age of all kinds, proteomics, metabolomics, what have you. And basically, one of the things we discovered then is that everybody ages differently. So just to show an example, this person, their top biochemical pathways that's changing is their cardiac hypertrophic signaling pathway, meaning this person's a cardioager. And we later learned they're stage 2 hypertensive. So it sort of makes sense, actually. This person over here is a typical ager. Their coagulation, metabolic, and other pathways increase with time. And so they're kind of a typical ager. We think you need as few as 5 measurements within 2 years. We can see how you're aging. And we've classified people into ageotypes. So every column is a different person. Every row is a different aging pattern, if you will. So the people over here, they're aging in all 4 categories: kidney, liver, metabolic, immune. They're more than 4. It's just that that's what we are statistically empowered to find. This person over here is aging in 3 of the 4, not a kidney ager, but aging the others. This person's kidney ager, kidney and metabolic, so on and so forth. So we're seeing how different people age. And then there are clinical markers associated with this. So the information is actually actionable. We've had people change their lifestyle and improve some of these markers. It's pretty cool. So bottom line is the combination of omics let us see how people are uniquely aging. Now, one question I get a lot is, that's great. That's a wonderful research study. How are you going to scale it? And the answer is, I'm in Silicon Valley, so we formed a company called QBio that's doing a medical version of what I just told you. But the most important thing out of this is they're doing, again, a deep data dive, but they've also added whole body MRI. Now, in today's world, most people tell you don't do whole body MRI because if you scan someone, you're going to find nodules. Men have them in the prostate, women in their ovaries, 100% of the time. And my point is, our point is that that's not the point. It's not the issue isn't whether you have nodules. The issue is, do you have growing nodules? And the way you find that is through longitudinal profiling. And so I can tell you, QBio, just from the first 100 plus people, found some pretty important things: early ovarian cancer, prostate, even found early stage pancreatic cancer, which is almost never detected early. And these days that can be life-saving. It's a big, big deal. If you have pancreatic cancer, you may know. So again, the deep data dive, in this case, more actionable data, has been very, very powerful for finding alterations in people's health. Now, one of the things we've wanted to do for a long, long time is try to actually bring this, if you will, into the home. We think the concept of going to a doctor when you're healthy, there are times when you're going to need it, no question. But much of the time, you can do a lot of these measurements at home where they actually might be more natural, more accurate, quite frankly. And so we've been pushing 2 fronts on this. One is the wearables, and the other is what we call microsampling, where you take tiny drops of blood and mail them in, and we measure thousands of molecules. I know what this sounds like. Ours does work. So the bottom line out of all this is that we have both these working pretty well. For the wearables, you may know there's over 1,000 devices out there, mostly in the form of watches and rings, blood glucose monitors, other things. They're super, super powerful because they measure you 24/7. They're measuring continuously. And that makes it easier to pick up when there are shifts in your baseline. And so one of the things we discovered early on when we put these on our cohort is that we could tell when people are getting infectious disease in advance of symptoms once again. And the way this came about is when I discovered my Lyme disease, of all things, from my smartwatch and my pulse ox. What we found was that I was helping my brother put up fences in rural Massachusetts. 2 weeks later, was flying to Norway, of all things, and I saw my blood oxygen was abnormally low on that flight. It's the blue one here. It should drop, but shouldn't drop that far. Never came back to normal when I landed. I also saw my heart rate was elevated, and I later learned my skin temperature was up too from my smartwatch. And this is all pre-symptomatically. And then later I got a fever off and on. I did go to a doctor after a few days. I warned him it might be Lyme because of the timing. Draws blood, sees my immune cells are up and says, yep, you've got bacterial infection. He wanted to give me penicillin. I said, no, I need doxycycline. Got a little tense, but he gave in. And it worked, cleared it right up. Got tests when I got back, Lyme positive. It was negative, given samples just before I left, so it was very fortuitous. Not completely random. I measure myself a lot. And so I was negative, so I seroconverted him. So we then went on to show we could tell when people are getting respiratory viral infections, actually from a smartwatch, again, pre-symptomatically. I won't show you the data for this. And we published all of this in 2017. And of course, as you might imagine now, what's happened is COVID came along and a couple of years later, and yeah, we're still in the midst of all this. Is another whole wave rolling out. And we wanted to see if we could actually use basically smartwatches to detect COVID. We think this could be a very, very powerful way, because if you think about the way we do it now with thermometers and stuff, that's ridiculous. Half the people don't get a fever, and it's not going to work. So we basically, so this was just showing that even advanced thermometers like these ones that, measure infrared, they still the same problem. A lot of people don't get a fever. So we basically, when the pandemic hit, we quickly partnered with Fitbit, quickly enrolled 5,300 people, of which 32 had COVID and had it while wearing the smartwatch, and had a diagnosis day and a symptom day. And the first step is, can we detect COVID with a smartwatch? And the answer is we could. This is our very first case. So you can see this is resting heart rate. This is their diagnosis date when they got COVID. This is their symptom day. And you'll see that their resting heart rate went up 9.5 days prior to symptom onset. So it really was an advance notice, if you will. They're probably spreading it around without knowing it because it was pre-symptomatic. And we went on, I'll show you in a minute, to write a real-time detection system for following this. And so it works. It worked 80% of the time. We could detect COVID at or in advance of symptoms. And the median was 4 days prior to symptom onset. So you can sign up for a study here if you like. So the point is, again, we think it's a very sensitive measure. So we've now set up a real-time detection system. It's a cloud-based system that can follow millions of people. Right now we have tens of thousands enrolled who are basically in the study, and it's tracking their mostly resting heart rate, but other things. And when we see an anomalous signal that's up high enough and long enough, it triggers a red alert. And it works, same number, 80% of the time. We basically find that we can pick up when people are getting COVID in advance or at the time of symptom onset. And here's again, one of our first examples. Here's when this person had symptoms. They were diagnosed the next day, but they were getting red alerts for 3 days prior to this. It works for Apple, it works for Fitbit. It works in not only pre-symptomatically, like I say, but it also works in asymptomatic cases. This is an example of this from a Fitbit. This person was diagnosed with COVID here, but they were getting red alerts for about 2 weeks prior to this, so easily could have acted upon that information. I want to emphasize it's not specific for COVID. Other stressors will trigger this. The number one stress of the red alerts is workplace stress. I know if we pull in more data types, we'll actually distinguish workplace stress from a COVID infection. That should be pretty straightforward. It's just that we don't have all the right data. It's hard to share. A lot of these smartwatch companies don't share their data at high resolution like we like. Other things trigger the stress too, including other infections. Here's a mycoplasma case. We even picked up influenza, what have you, will trigger these. Some of the stuff, it's easy to contextualize. Like if you're traveling and you get a red alert, that's there could be reasons for that stress, or if you're in an airplane, the blood oxygen drops, your heart rate goes up. So that's another thing. All right. I can tell you it's actually even more sensitive. I won't have time to go through this in detail, but it's more sensitive than, in fact, a COVID antigen test. And here's an example. This is my example, but we have others as well. In this case, what happened was it was just a touch congested. I wasn't sure if I was coming down with something or it was allergies, what was going on. So I did a COVID antigen test. It was negative. And then I did my smartwatch, looked at my smartwatch. It was positive. I'd had some red alerts prior to that from a particular family stress event. But anyway, I was positive while my antigen test was negative. And once you know it, I actually traveled to New York City, had to get tested the next day to go to a meeting. And sure enough, I was bright positive. And so the net result was that I spent a whole week in New York City, all because I listened to my antigen test and not my smartwatch. Again, so my smartwatch is more sensitive, can tell when something's going off. It's not as specific. I didn't know it was COVID, but I did know something was up. And in hindsight, I don't make that mistake twice. I will tell you that we think this is just the tip of the iceberg. You can get a lot of information from a smartwatch for health monitoring. And we use machine learning these days and AI to be able to measure things that are, signals from smartwatch. And we can get rough measurements of hematocrit, red blood cell count, hemoglobin, all from a smartwatch. And if you think about the way they work, this actually makes a lot of sense. You're shining light into your blood. It reflects back, and you can actually get these kinds of measurements. It even can pick up hemoglobin, A1C, fasting glucose, what have you. Again, not clinical grade measurements, but certainly good enough to see shifts from baseline. Other kinds of wearables that are powerful, you may know, are glucose monitors these days. So they were being used at the time for insulin-dependent type 1 and type 2 diabetics. When we started this, our claim to fame was we started putting these on basically normal people, and people are pre-diabetic. And what we discovered is that some people are normal, actually are pretty good, have good glucose control, don't spike much. Many people are moderate spikers, quite a few. It turns out there's also some who are severe spikers. They're not as normal as they think they are. In fact, they're spiking just as bad as diabetics. And so we wrote algorithms to classify people into glucotypes at low, medium, high. We're actually doing a lot of work. Well, I'll tell you in a minute. So what we found is that different people spike to different foods. This is some work from Aaron Siegel, who found the same thing. Some people spike to bananas, others potatoes, others to pasta. It really varies from one person to the next. And so what we discovered is that, here's his work. Here's a person who spikes a banana, but not a cookie. Probably the only person on the planet doesn't spike to a cookie. This person spikes a cookie, not a banana. That's a lot more typical. So anyway, you get the idea. Different people spike to different foods. And we're doing a lot of studies these days of trying to understand nutrition and how it affects people's metabolic health, including glucose spiking. I will tell you, this is one thing we learned is that 80% of people spike to cornflakes and milk. This stuff is nasty, probably worse than smoking. So if you don't remember anything else from my talk, remember this, watch out for cornflakes and milk. Okay. The other thing we've been doing a lot on, and this is very relevant, I think, to the proteomics, I think this group will like this, is this remote monitoring and this idea, why do you go to a doctor when you're healthy to get a lot of these measurements? Now, some make sense. You're not going to do a whole body MRI at home, but many measurements can be done right at home. And there are certain situations where it's very advantageous to make these measurements, or at least samplings at home. So we spent about 6.5 years working on this, doing collecting little drops of blood. You FedEx them into our lab, and then we do lipidomics, metabolomics, proteomics. We do these deep data dives. We measure over 2,200 analytes. We even have targeted assays against key molecules like cytokines and cortisols and interesting hormones. And so what we discovered is that proteins are actually pretty stable. Metabolites are mostly stable as well. Lipids, a little bit less so. It depends on the lipids. But we know which ones are and which ones aren't. We can correct for that if needed. So one of the cool results we discovered, we after setting up this assay, is we did some fun studies. This is one of them. We had 32 people drinking an Ensure shake. And we measure them before drinking the shake, then at 30 minutes, 60, 120, 240 minutes. So a very common shake you find over here in the US in drugstores, et cetera. And what we discovered is basically that, this is not a surprise. Over 1,000 molecules change after drinking the shake when they were fasted initially. And we can measure key molecules like C-peptides from insulin itself. This is an incretin, other things. We can actually quantify exactly what's going on with their hormones after drinking this. The cool result was this slide here. Everybody reacted to the shake differently. This is just showing carbohydrates, 3 of them. They all behave pretty much the same. Each block is a different person, and this is a time course. This person, their carbs plummet, relatively speaking, compared to other molecules. This person skyrocket. About the same. This person's goes down. This person's goes down. So different people are reacting to the shake differently. We group people into 5 different groups. Gray is the average here, and this is essentially some signatures. This one's really pretty cool. That this brown ball is inflammatory markers. Turns out in this group of people, their inflammation markers plummet. In this group of people, they go up. So this group of people, they go up. So the same shake is having different effects on different people. In some people, it's anti-inflammatory. In others, it's pro-inflammatory. And we can measure this. That's the key. So we can see exactly what foods are doing what to which people. And so since 10% of the US has inflammatory bowel syndrome, we think this kind of information can be very, very powerful for seeing what kinds of foods inflame which people and therefore take mitigation measures. The other thing we've been doing is this here. This is the one that puts it all together. We've basically been doing, this multi-omic sampling on one person very, very deeply. Every waking hour, they gave a sample for basically 7 days, and they're wearing a CGM, a smartwatch, and doing food logging. So we can start correlating physiology, biochemistry, and people's activities all in the same person. And then we look for correlations between their biochemistry and physiology and so on and so forth. And what we discovered is shown here. Basically, we found thousands of associations between people's, say, activity and physiology and their biochemistry. So here's heart rate, glucose, and steps. We can correlate with the cytokines. These are these purple metabolite or lipids, are blue, and metabolites are in green. And some of the stuff we see makes a lot of sense. We'll find, for example, this glucose C-peptide goes up after glucose that you would expect, but we can quantify that in this person. It's exactly 10 minutes. Here's a new immune marker that went up in response to glucose. That was kind of unexpected. It might actually be responsible for why people get inflammation when they're associated with diabetes and high glucose. This one turned out to be really cool. This is a marker that's been implicated in dementia and Parkinson's, and we discovered it does correlate with glucose. Believe it or not, that was known. But the surprise was that we can actually see it's showing very interesting patterns that we think correlate with stress. So imagine you're at risk for dementia or Parkinson's, you probably would want to, engage in lifestyles that would not spike this, that could lead to these aggregates that cause dementia and such. So the point is that we think this could be very, very actionable information for actually helping prevent, improve mental health in the long run. So the bottom line is we've set up a dashboard to bring in all these data types. So we can collect wearable data, omics data, clinical data, bring it all in onto a common dashboard. And then we can display it at whatever resolution you think is useful, daily, weekly, monthly, what have you. So the idea is to empower the individual to be able to better manage their own health, because a physician doesn't have enough time to do this. So, but we think if you can empower people to do this, and it will have much, much more effect. I can tell you this is where we like to see the field go. We're believers that all this data can be very powerful for managing and health and transforming the healthcare system. We're believers in the future that people will get their genome sequenced prior to birth, Then together with omics and personal, wearable sensing, you can better track people's health and better manage their health, and then ultimately build these AI-driven management systems for essentially doing just that. We have, again, because I'm in Silicon Valley, the way we try to scale this is spinning off companies. So I founded 17 over the years for things, and this is one involved in metabolic health, same with January Eye. This is a CGM company, very interesting, that's actually built a whole health management system around CGM. This one actually lets you do remote monitoring where you can, do just that. You actually do a little prick of blood. It's based on our microsampling, mail in a kit, get back data on, in that case, about 500 molecules. We have a long COVID company. It's also doing the microsampling, wearables, and so on and so forth. So we think we try to translate what we're doing to a broader group. I have an amazing team that's been actually leading this effort. So this is the wearables team when it was at its peak. It was the microsampling group that's out there now that's been helping actually drive a lot of this. I do want to give some shout outs who's not here, but one of the key drivers of a lot of this was Wenyu Xiao and Rui Chen, who actually set up a lot of this IPOP system. And I will say that it turned out to be a very valuable cohort. We've used them for actually tracking the effects of exercise, nutrition, so on and so forth. So we've really learned a lot from the study and the deep data profiling. So I should have left plenty of time for questions. I'm happy to take some. So if any of you have any, please ask them now.
Thank you very much, Dr. Snyder. That was a fantastic talk to kick off the sessions today. It's truly fascinating to witness how the ongoing advancements in technology are propelling this transformative change in healthcare. We will now be moving to the Q&A section of this session. If you would like to ask Dr. Snyder any questions, please submit them to the chat system to the right. And as we delve into the fascinating world of health monitoring using big data, it's crucial to address one of the most pressing concerns, privacy. Dr. Snyder, can you shed some light about privacy concerns and what are the ethical and privacy considerations surrounding the collection and use of personal health data for deep data analysis and remote monitoring?
Yeah, great question. So a lot of people are concerned about privacy. And I think they're worried about getting discriminated against by their healthcare system they're in. In the US, you actually can't be discriminated for health care or for employment based on, say, your genome sequence. You can for long-term disability and life insurance, but other kinds of data haven't really been dealt in with this. My own view, though, about the privacy in general is I'd say get over it. We just, we're not in a very private world in general, meaning many of you have credit cards, and that has a lot of personal information. We do share that with a credit card company. We do that because it's very convenient when nobody wants to walk around bags of cash. So we think, you will sacrifice certain things to be able to live a better life. And I think that's true of this health data. I think it can be used for good as opposed to bad. And so I think if anyone is going to abuse it, I do believe that everyone is entitled to some level of health care and that we should be certainly making that happen as best as possible. And so I would argue that for, if people are getting abused by the system, then we need laws to protect them and ensure that everyone gets access to, again, some level of health care. It may not be Cadillac health care, but basic health care, I think we should all be able to, we're all entitled to.
And isn't this too much data to make sense of?
Yeah, I think the answer is no. I know it seems overwhelming. As you mentioned at the outset, I had 2 petabytes of data just on me, which is true. And I forgot to say, but I'm wearing these 4 smartwatches, and you see my Aura ring off, and wear a glucose monitor, and so on and so forth. So it's a ton of data. Now, this is a research study. We're trying to basically understand, what data types are most valuable, what resolution we should be collecting data, all this sort of stuff. But as we roll this out, I think it can be synthesized into a simple form much like your car. So your car actually has a lot of sensors on it. Race car has as much as 400 sensors on it. And yet you don't have 400 monitors displaying on the screen. You just have a few because it synthesizes the data and brings it back and shows you the most important information. And it can be simplified, like a check engine light tells you something's off, doesn't tell you what's off exactly, but it tells you when something's off so you can go do follow-up to test it. So that's how I see health monitoring in the future, that we can collect these data that get synthesized. And to be honest, AI-based programs can actually basically make personalized programs. They'll understand your baseline, your patterns, and then detect shifts from that and actually better monitor your health. And I hope catch disease, even tell you what kind of disease you have. I'm hopeful the smartwatch, for example, I'm pretty confident we can tell the difference between mental health and, as I mentioned, and a respiratory viral infection. We just need the right data types to do that. So I think the big data really helps us distinguish more precisely what's going on. And so we're big believers. It can be very, very powerful.
Thank you, Dr. Snyder. Could you elaborate on other challenges that may present with implementing using big data and remote monitoring into practice?
Yeah, another great question. One of the biggest challenges in the US is who pays. Nobody pays to keep you healthy. To be honest, the single payer systems have a better chance. Places like Canada and Europe, for sure, have a better chance of implementing this than the US does, because US system's so fragmented. And again, no one pays to keep you healthy. So people have to pay out of pocket. So the only way this is really going to get truly implemented is to show it saves money, saves the payers money, etc. My own view is that, and then there's also getting people to comply with this. That's another challenge. Imagine you could implement this. Would people do it? And I think the answer is they would do it under two conditions. One is that you actually pay them or give them discounts, or you get their family involved. So I'm a believer you should actually get a discount in your healthcare and your insurance if you wear a smartwatch, because you're going to better manage your health. Same with your genome sequence. You should actually get paid to get your genome sequenced. So I think we need to realign our financial models to get them properly aligned with the existing system, so we can indeed better manage people's health. Yeah.
Thank you. As we are now drawing to a close for this session, I do have one final question to ask. You briefly just touched on this from your previous answer, but can you just build on a little further? What are the potential economic implications of incorporating deep data and remote monitoring into healthcare systems in terms of cost savings and resource allocation?
Yeah, great question. So my own view is that obviously the research study we did cost millions, super, super expensive. And QBio, it costs $3,500. Now, I would argue that it's worth it, because if you catch, those early cancers and things and early heart conditions, you're going to save a lot of money. So I think in some spaces, there are economic models that really make this obvious, like in the cardiovascular risk space. If you're at high risk for cardiovascular disease, to get health monitoring, it's going to save you, because if you get a stroke and wind up in long-term disability, that's very, very expensive. So I think the cardiovascular space, you can make good arguments for genome sequencing and tracking. I think it's true of other areas as well. Certain kinds of technologies are going to be dirt cheap to roll out, like the wearables and such. We just got to get them approved and show utility. But I think these are incredibly inexpensive devices. These days, the expensive ones are $250, and they measure a lot of stuff. So, but for $50, you could actually do pretty much everything I just told you, a $50 smartwatch. So imagine having health tracking for, a large fraction of the population. Imagine you can do it for half the world. Half the world has a smartphone, you may or may not realize. And then if you could team that with a smartwatch, you have a health monitoring system for a lot of the world, and you would actually have then an infectious disease tracking, because you could see if people's temperatures are going up and see that in advance of symptoms. You could have an early warning. I think it would save billions of dollars. I think the wearables will pay for itself very, very quickly. Other areas that may take time, the prices are dropping. like I said, the genome sequencing, we pay $500 for a human genome now. It's going to be $100 by the end of the year. It's pretty amazing. So I think it will, even though it starts out expensive, it actually will be inexpensive, and I think it has the potential to save a ton of money for the healthcare system.
Thank you so much, Dr. Snyder. That concludes our Q&A session. And before we wrap up, I'd like to remind everyone watching that any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Snyder. It has been an absolute delight having you open this year's Advances in Proteomics and Metabolomics online symposium. For all our viewers, stay tuned as we'll return shortly with the next talk.
Thank you.